Okay. You should have heard this meeting is being recorded. Okay, perfect. So hello, everyone. Thank you for joining us for the first microTAStic seminar of 2023. For those of you joining us for the first time, this is a new virtual seminar series running the first Friday of each month from December through May. And we are sponsored by CBMS. We are grateful to their assistance to reaching the international microfluidics community. So just as an overview, these are the seminars we have coming up here in the future. And, today we're really excited to have Shanna, Kelley, and Tejal Desai. If you want to get more information about the seminars that are coming up or be added to the mailing list to receive the Zoom links, you can do so at this microTAStic website here. I'm going to let Shanna begin sharing while I give her intro. So we're, as I mentioned, very excited to have two fantastic speakers today. So first up, we have Shanna Kelley, who is professor of biomedical engineering and chemistry at Northwestern. Her research interests are on development of new bioanalytical capabilities, and she's an innovator with over 50 issued patents and is founder of 4 life science startups. She's received numerous awards, but to name a few, she's a fellow of the American Institute for Medical and Biological Engineering. She received the ACS Nanoscience Award, and she's a fellow of the Royal Society of Canada. So without further ado, I'll turn it over to Shanna.
Thank you so much. It's great to be here today. I really appreciate the invitation. And so I'm going to talk to you today about one of my group's technology platforms and tell you about some applications that we've developed. Just quick disclosures on a couple companies that we've spun out of the labs recently. I'll tell you a little bit more about Control Therapeutics at the end of this talk. And I'll just start off by introducing my group if you haven't come across us before. I'm a bioanalytical chemist by training, and over the years we've developed many different analytical technologies that allow us to make very sensitive measurements of biological systems. And we do this through 2 main modalities. We do a lot of electrochemical sensing to develop sensors that can be used for clinical diagnostics. And then increasingly, we are focused on wearable sensors, implantable sensors, things that allow us to monitor physiology in real time. The other big area of effort in the group is in rare cell profiling. And that's what I'm going to spend my time on today. We've had some really interesting new developments with this platform that I'll describe to you. And I thought for this audience interested in microtechnologies that this would be a good one to spend the time on today. And I don't have a huge amount of time, so I'm not going to give a very long introduction. But I will just say a few words about the rationale behind the creation of the platform I'm going to tell you about. I think we'd all agree that single cell analysis has really revolutionized biology, right? We used to really only have the capability to look at bulk properties of cells or ensembles of cells. Now, the fact that we can go cell by cell and measure biomolecular expression, measure genetics has really transformed how we can understand biology. And it's certainly had a huge impact in the areas here and beyond. But I think, those of us who work with single cell analysis technologies, we also know that there are limitations, right? It's very difficult to look at large collections of cells. And there are some other things that are limitations. And what this means is that there are applications that are difficult to get at with existing single cell technology. For example, if you want to hone in on very rare types of tumor cells, those can get lost in the background. If you want to look at the immune cell complement and try to identify tumor reactive T cells, that can be a challenge. And then we've also done some work that I won't really have time to talk about today, looking at large collections of CRISPR edited cells to be able to pick out interesting phenotypes. So there's certainly lots more to do here. And so, we really focus in on rare cells, looking at rare cells at the single cell level. And looking at rare cells, it's a needle in a haystack problem. And looking at a haystack is not very efficient if you look at one piece of hay at a time, right? That's a serial process. It's really slow and it's difficult to work with, the billions of cells, let's say we need to look at if we're analyzing a human blood sample. And so speed is definitely a bottleneck of many different single cell processes or measurement technologies. And then there's also the signal to noise problem, right? If you have a vast background of non-target or uninteresting cells, and then just maybe 10 or 20 or 50 cells that you want to look at and measure, the signal to noise just drifts in a way that it's not particularly informative. So these are some of the challenges. And, one technology that has been incredibly powerful for looking at cells at the single cell level is FACS, so fluorescence assisted cell sorting, or if we're not doing it preparatively, we're just doing an analytical measurement, we refer to it as flow cytometry. Here you take a fluorescently labeled antibody, put it into a mixture of cells, and then pass those cells single file through a cytometer that then assesses the level of fluorescence on a cell, right? And that comes from the antibody accumulating on the surface of the cell or inside of the cell. And then, one cell at a time, sorting those cells, providing a readout of the levels of fluorescence. And again, this is very powerful, but there are throughput limitations because it is one cell at a time. There's also recovery limitations. You're also squeezing cells through an aperture that is the size of a single cell, and that induces stress and other issues that make it such that you don't get all of the cells back that may have been in a sample. So this is, a workhorse technique for many of us, but again, does we could use new tools. So that was the rationale for developing this technology platform that I'll describe to you that we refer to as magnetic ranking cytometry. And so here, rather than using a fluorescently labeled antibody, we use very small magnetic particles as our label. So we take the particles, we put an antibody on the surface of the particles, or it can be another type of receptor, an aptamer or other things. We put those particles into a mixture of cells. It can be whole blood, it can be a dissociated tumor. And then those cells get passed through a microfluidic device that has a variable depth. We can create gradients other ways, but in this particular example that I'm showing you here, we create a depth gradient. And so then what happens is that the cells that are differentially labeled pass through this device and they get deposited in different zones of the device depending on levels of magnetic labeling. So we process a sample, the cells kind of settle in these different zones according to how many particles they're carrying. And then if we want to do an analytical measurement, we can scan across the channel. We come in with a cocktail of immunofluorescence stains or other types of chemical stains. We scan across the channel, and then we create these histograms showing the single cell level data, where each cell landed in this channel. And that is a measure of protein expression. So we get this type of histogram where we can see these distributions. That allows us to measure protein expression for an ensemble of cells. And it also allows us to do it at the single cell level. And one of the things that was critical in getting this to work was really coming up with an approach that allowed us to trap the cells, because these magnetic particles do not give you a lot of magnetic force to work with. So the cells are actually hard to trap, and we had to then develop these X-shaped structures that allow us to create regions of low flow within the device. And so I'll just show you a video of this is whole blood flowing through one of these devices. You can see there's a pile of cells that are accumulating. in this low flow zone created by the X-shaped structure. So here we just have a little chamber. It has magnets above and below. It's quite small, size of a penny, and then the cells start accumulating. And so this is how we're able to kind of get them out of the complexity of whole blood, and then pattern those X's along a channel so that we can do the measurement. And so once we process a sample, those cells that have collected in that low flow zone, you can see that they're, right here staying put because they have enough magnetic particles to resist the pretty low drag force in that region of the device. And then once we've processed a sample and then scanned along the channel to see where the cells settled, we can create these histograms that I showed you a moment ago. Here we're looking at the protein EpCAM, which you find on the surface of epithelial cells. And so here we have 3 different cell lines with 4 different cell lines with 4 different levels of EpCAM, which you can see by flow cytometry, which is in the inset here. And then, we can visualize levels of protein expression and get very similar results as one can achieve with flow cytometry, but using many fewer cells. So 100 cells, and we get really nice looking histograms and distributions. If we want to multiplex, this is one limitation of the technology, that we don't have 10 different colors of fluorescence. We just have kind of one color of magnetism. So if we want to profile multiple markers, we have to aliquot a sample and then do measurements on each aliquot. So then, that's maybe a limitation, but where this technology really does something that is very unique is that it can find 10 cells or 20 cells or 40 cells in a milliliter of whole blood, where there are billions of cells that are really just background. And we can quantitate protein expression, measure it, and that expression measurement stays very constant, irrespective of the cell count. And so we have done this. We originally developed this approach looking at surface proteins on the surface of a cell. We then later figured out how to fix and permeabilize the cells. Now the cells obviously are not alive, but we can send the particles inside to label intracellular targets like RNA molecules or intracellular proteins. And so we've done a lot over the years with really adapting this capability and making it amenable to the measurement of lots of different targets. So then we've also taken the same approach and made it preparative. So what I've just showed you was an approach that allows you to do analytical measurements, measuring protein expression or expression of other biomolecules. We've also adapted this so that you can process a cell mixture and then walk away with the cells, the subpopulation of cells that are of interest. So same basic device architecture where we have a linear velocity gradient, and we're doing magnetic trapping based on levels of labeling. But here we have discrete chambers, and so we can process a sample. This particular example, we have 4 chambers, but we can have 10 or we can have 100 depending on the needed resolution of the separation. And we can walk away with those cells, expand them, sequence them, characterize them with very, very high levels of throughput. So billions of cells an hour can be processed through a single device. Recovery levels are very high and viability levels are very high. And these are the things that really differentiate this type of approach versus, let's say, flow cytometry. And just recently, we've been working with the devices that I just showed you, but also with deflection devices where we have arrays of magnetic guides. And then instead of using those X-shaped structures, we kind of deflect the cells according to levels of magnetic labeling. And what we've done recently is to package up arrays of magnetic guides, 3D print those in a cartridge, and really then up the throughput further. And this was just published in Lab on a Chip, and it's a nice fabrication method that we hope will allow us to roll this out more broadly. So that's the basic technology platform and the approach. What I want to do now is just walk you through some of the applications that we've developed using this platform. We've used it for a lot of different things over the years, and we've really honed in on problems where you need to look at billions of cells at a time and have that level of throughput. And one area where that's certainly the case is in liquid biopsy. And here I'm referring to finding very rare cells in the blood and being able to say something about protein expression levels. We've done that for rare tumor cells in the blood, also immune cells in the blood. Another kind of related area that is pretty recent for us is looking at extracellular vesicles in the blood. So we've adapted the platform, made it amenable to working with those very small particles that you see being released out of cells that are a really interesting class of biomarkers. And then we've also started using this platform to do selection. So one area is in the selection of antibodies. So these are phage displayed antibodies. We put those into a complex collection of cells, and then we pull out very specifically the cells that we're most interested in finding binders to. And the complexity and the ability to do the on-cell selection gives you very high specificity binders. And we showed that for a surface protein called Frizzle7 just earlier this year. And then we've also used this to do aptamer selection. So using particles that are displaying aptamers on their surfaces, and then putting them through this type of device, doing labeling so that we can pick off aptamers that actually have programmable biophysical properties, whether it be KD or K on or K off. And so that's, I think, a really interesting new application that we've just kind of finished up a proof of concept on. We also do a lot of work on therapeutic target discovery. So we use this approach to sort through libraries of CRISPR edited cells. And so there we're looking for interesting phenotypes that are related to the progression or the origin of disease. And so we can pick out interesting phenotypes, look at the knockouts that are present in the cells that are maybe overexpressing a cancer-related protein or have lower levels of expression. And by doing that, it gives us a high throughput way to discover new targets for therapeutics. And so we have a variety of studies that we've published recently and quite a few more to come on that. But where I'm going to spend the rest of my time today, and I know I don't have a huge amount of time left, but I'm just going to talk to you quickly about some of our work in the area of cell therapy development. And so here, sorry, my animation's a little off there. Cell therapy is an area that I'm personally really excited about. If in the context of oncology and being able to treat very advanced cancers, I'll show you that there are examples of cell therapies actually being curative for patients that really don't have any other options left. And that potential is very different from what we see with our molecular drugs, right? Where we typically see a few months, extension of life. But with cells, because these are living therapeutics, again, they have the potential to cure disease. But there are problems, right? The cost is very high. The manufacturing is very complicated, and the logistics are difficult to integrate with mainstream clinical medicine. So this is an area that we started working in kind of over the pandemic, decided to learn something new, dive into tumor immunology. And we now have a couple papers, one's out, one's kind of probably going to be out in the next couple of months in this area. And so I'll just take you through two, very short stories in the interest of time. And they're both focused in this area of tumor infiltrating lymphocytes. And if you've never heard of that particular type of cell therapy, I'll tell you what it's all about. But just quickly, how these therapies work. So in the instance of oncology, there are many different types of cells that are being deployed to cure cancer, but most commonly, these are T cells. And with T cells, just understanding the therapeutic activity, it's really related to two things. So you have the recognition of a tumor cell by a T cell, And they do that using their TCRs. And then the T cells also are able to be cytotoxic. Once they've recognized a cell, they can then secrete things that will kill tumor cells. And our immune systems are always surveilling, right, our tissues and looking for these kinds of cellular aberrations. And in cell therapy, we're just really trying to kind of amp that up to allow the immune system to eradicate tumor cells more efficiently. So then in TIL therapy, I mean, there are many different approaches that are being contemplated for cell therapy in cancer. There are things like CAR T, where you engineer a cancer recognizing construct into a T cell. But today I'm going to talk about a non-engineered type of cell therapy, which is TIL therapy. So the idea behind TIL therapy is that we know that in tumors, there are immune cells that have infiltrated the tumor tissue and are trying to eradicate it, but they become overwhelmed, right? But the idea behind using this as a therapeutic is if we could isolate those cells out of tumor tissue and then expand them ex vivo, those cells can then be brought back into a patient to eradicate more of the tumor cells. And this was an idea that was really championed and pioneered by Steven Rosenberg at the NCI, decades ago. And TILs are now really getting momentum in the clinic, and they've been shown in a small number of cases, but a significant number of cases, to be curative for things like metastatic melanoma. This is really the only type of cell therapy right now that's effective against solid tumors. And one of the key things is that they're in TILs, you're harvesting all of these T cells. You have great diversity in the TCRs, and so you're able to eradicate multiple different types of tumor cells within a tumor, and that really helps with resistance. So, again, there's a lot of good data out there, but it is mixed. So when you look at the clinical trials that have been run, for example, on melanoma, and you look at patient response, there's in a study typically of a few complete responses, lots of partial responses, but it really varies across the patient population. And there's also been this bottleneck of characterizing doses of TILs. A dose of TILs is 50 billion cells. And so now I'll come back to magnetic ranking cytometry and how we've deployed this against this problem. But you can see that the cell count is very, very high. And that's why we need really high efficiency cell processing technologies. And just to give you an idea of how TILs are manufactured, so the first step is a surgical rescission of tumor tissue. So you take the tumor tissue out of the patient, gets shipped off to a manufacturing facility, gets fragmented. The T cells are cultured out of the tumor. And then once you have a culture that's mainly T cells, that goes into a rapid expansion protocol. And then the doses of cells are cryopreserved, brought back to the clinical site, infused into a patient. This takes a while. It's pretty complicated. And if you think about what's in a dose of TILs, those 50 billion TILs you have cells that are cytotoxic and tumor reactive, but you also have bystander T cells that are not yet tumor reactive. Those are very fresh. They're typically naive cells and they grow really well in culture. So there can be a large number of those in a dose of TILs, and then also exhausted cells that don't have any more cytotoxicity to give. So we thought this was a really interesting application for magnetic ranking cytometry. And a really talented graduate student, now postdoc in my group, Daniel Wang, decided to go after trying to sort doses of TILs to find the most reactive ones. And he used a marker called CD39. This is a marker, I won't go into the biology, but it's a marker that you find on T cells that are tumor reactive. But if it's at very high levels, those T cells are also exhausted. So Daniel's idea was that he was going to take the moderate expressors of CD39 to see if they had differential activity as an anti-tumor therapy. So Daniel, went after, he adapted the platform, and I won't go into the details of this. It was published earlier this year, last year. And so he did this in a mouse model of colon cancer. He isolated the CD39 moderate expressing cells. You can see they're a smaller subpopulation than the highs, which are the exhausted cells, the lows, which are the naive cells. And then he characterized these, and you can see that the phenotypes really kind of behave as you might expect they would, with stemness being very low in the exhausted high cells. And then some of these other phenotypic properties, trending the way that you would imagine based on this kind of tumor reactivity, exhaustion type of behavior that I mentioned to you. But then the real test was he took these different subpopulations of cells, he expanded them, he injected those into a new cohort of mice, and then looked at anti-tumor activity. And you can see that the CD39 mediums are the most potent cells. They give you a much more dramatic anti-tumor effect versus the highs or the mediums. So that shows that there's really this sweet spot in a dose of TILs that you can get to with high efficiency cell sorting. And he also compared to just unsorted TILs. So the trials that have been done in the clinic are with unsorted TILs. And you can see that the effect is actually pretty small, much more pronounced with the CD39 medium cells. So this was a really nice kind of first proof of concept study applying our technology to cell therapy development. And just in a couple minutes, I know I'm pretty much out of time, but I'll just talk to you about kind of the next phase of this work that has taken us to a really exciting, I think, new direction. And what we wanted to do was to kind of get away from the invasiveness of TILs. So to harvest TILs, you have to start by doing surgery. And Daniel Wang, same student I just mentioned, started to think about whether TILs might move from one tumor to another through the bloodstream. And so he did a variety of experiments. I won't go through it again. The paper is going to be out in a couple of months, and I'm happy to send around preprints. He was able to prove that TILs do move. He then went and looked for them in the bloodstream using the magnetic ranking cytometry platform, which is really good at finding rare cells. He found these TILs in the bloodstream and then was able to sequence them to find markers that we could use to isolate these cells, first out of animals, and we've now done some good proof of concept in humans. So he discovered these markers, he validated the markers, and then went into, again, colon tumor model, isolated these cells, which we call circulating tumor reactive lymphocytes. And in looking at anti-tumor activity, he showed that these cells do have a high level of anti-tumor activity versus the cells that don't have the markers. There's a high durability of the response, the cells stay around. And then he's also done TCR sequencing to show that the cells that we find in the blood are a subset of the cells that you find in tumor tissue. So these are kind of one and the same cells. And then finally, just very quickly, he's also looked in human specimens for these cells, and one can visualize them using our platform. We've looked at colon cancer patients, lung, breast. We've also done some mesothelioma, and you see those cells across all of these different tumor types. So this is really exciting, the potential to maybe deliver a response like what one sees with TILs, but with a selected subpopulation of cells for a high quality product, and also a modality that can be used without the need for surgical resection of tumor tissue. So we do have a company now that's been kicked off and funded. It's here in Chicago. It's also has an outpost in Toronto, and we're kind of off to the races with trying to develop this type of therapy as a new solution for the treatment of solid tumors. So with that, I'll just wrap up. I've told you about this one application of magnetic ranking cytometry, but lots of other exciting things going on. I want to again recognize Daniel for his extraordinary work on the science that I just described to you. Randy and Mahmoud are two research professors with us that work on other aspects of magnetic ranking cytometry. I thank everybody in the group for all of their great efforts, as well as our financial supporters. And also, if anyone's looking for a new opportunity, a postdoc, you want to come and join us as a research professor, I'd love to hear from you. We're recruiting at all levels. So thank you, and sorry for going a little bit, over close to time. Thank you.
Thank you, Shanna, for the fantastic talk. So I think in the interest of time, I'll go ahead and just ask the first two questions that came up in the chat during your talk. So the first one is whether you have accidental capture of non-target cells.
We do to differing degrees. So we have these different device architectures that help us with that. If we need very high purity cell subpopulations, we typically use those deflection devices because you're not capturing in compartments, but instead the cells fall off of the guides. It gives you a higher purity fraction. So we struggle with that the same way others do, but just depending on how critical it is that the purity is very high, we have ways of dealing with it.
Great. And then the second question was whether or how you mitigate clotting in the device, especially if you have blood clots in the high shear regions of the device.
Yeah, so our devices, they're actually more macro than micro, right? We don't have very small channels. So debris, what we've seen, it tends to just kind of make its way through the device. So that is, it hasn't been a huge issue for us, but it's mainly because of the dimensions. The channel is quite wide. It's pretty deep, relatively speaking. So not as much of an issue for us as for technologies where you're down kind of more at the micro scale.
Okay, great. And so I think we'll turn it over to Tejal now. Shanna has kindly agreed to stay on a little bit after the talk today to answer additional questions. So for those burning questions that we didn't get to, please just stay on afterwards. And thanks again, Shanna.